Hello students, welcome back to Mathsci Educator. Today we are going to learn our 12th standard biology and we will continue with the chapter number 12, that is biotechnology and its application. In this chapter we have already completed about the biotechnological application in agriculture and now we move towards the biotechnological application in medicines. Then after we show the transgenic animals and ethical issues. So now we take about a biotechnological application in medicines. The recombinant DNA technology process have made an immense impact in the area of healthcare by enabling the mass production and of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not include unwanted immunological response as is common in case of the similar product isolated from non-human resources. मतलब कि जो non-human resources में से जब हम isolate किसी भी medicinal product को अगर हम isolate करते हैं, they create a somewhat allergic reactions or something that side effects. So, by using such type of the recombinant therapeutic approach that eliminate an unwanted immunological response because these products are isolated from a human sources. Right now, at present, about 30 recombinant therapeutic have been approved for human use worldwide, and in India, 12 of these are presently being marketed. Among these, today we are learn about a genetically engineered insulin. As you know, what is the use of insulin? They are used as an diabetic product. मतलब डायबिटीज जिसको होती है उसके क्योर के लिए. So this one is the cure for a diabetic patients. A management of adult onset diabetes is possible by taking a insulin at a regular time interval. Nowadays, this insulin can be obtained from a bacterium using the technique of biotechnology. But previously, they are extracted or maybe isolated from the pancreas of slaughtered cattle and pigs. But insulin from this source is they develop a allergy or other type of the reaction to the foreign product. To overcome and another things, that is the insulin consists of two short polypeptide chain A and chain B, and that are linked together by a disulfide bond. As you seen in this figure, in human insulin is synthesized as a prohormone or a prohormones like proenzymes, which contain an extra strange known as a C-peptide. As you seen in this figure, this one is the proinsulin. This blue color, that is the peptide 1, and this green color that shown a peptide B. Okay, दो तरीके के यहाँ पे पेप्टाइड है, and these two peptides that bond with a disulfide bond and create a insulin, and then after that free the peptide C. तो ये जो A पेप्टाइड है और B पेप्टाइड है वो दोनों एक दूसरे के साथ यूजिंग दिस डाइसल्फाइड बॉन्ड दे create or produce the active insulin. The main challenge for the production of this insulin using the recombinant DNA technique was to getting the insulin assembled in a mature form. Now, as you seen in this figure, an American company, Eli Lilly, in a year 1983 that prepared a 2 DNA sequence that corresponding to chain A and chain B that are identical to human insulin and introduced them in a plasmid of E. coli to produce the insulin chain. As you seen in this figure, this one is the first plasmid in which insulin gene that produce a subunit is incorporated or recombinant. Okay, तो इस प्लाज्मिड में उन्होंने recombinant किया है insulin gene that produce a subunit. Okay, now in this plasmid that recombined insulin gene that produce B subunit. What they do? They first isolate A subunit producing gene and B subunit producing gene. Then incorporate both these A subunit and B subunit in an individual plasmid. Then transfer that plasmid that produce A subunit and B subunit in a different E. coli cell that transform this E. coli with a recombinant expression vector and select the transgenic cell by their antibiotic resistance that we already learned in about previous lecture, in about previous chapter. कि हम इसका स्क्रीनिंग किस तरह से करेंगे, बाय यूजिंग द एंटीबायोटिक रेजिस्टेंस जीन. तो यहाँ पे उन्होंने किया है कि ए सब यूनिट को आइसोलेट करके उसको एक प्लाज्मिड में इनकॉर्पोरेट किया है in which they create antibiotic resistance gene, and because of that this plasmid get incorporated into the E. coli cell. Same wise they isolated insulin gene that produce a B subunit and create and plasmid and transform them into the another cell of Escherichia coli. Now this Escherichia coli or E. coli, they get cultured and produced these beta subunit and A subunit in their productive culture. Then after they purify the fusion protein. उन्होंने क्या किया, ये जो प्रोटीन जो जो कल्चर मीडिया है उसमें जो प्रोड्यूस हुआ है उस प्रोटीन को उन्होंने प्योरीफाई कर लिया फ्रॉम द कल्चर एंड केमिकली रिमूव the insulin polypeptide from the beta galactosidase protein. Then after they chemically remove this insulin polypeptide from the beta galactosidase. Now they combine this polypeptide to produce the functional insulin that is as same as a human insulin. उन्होंने by using a disulfide bridge. Okay. देखो आपको जैसे इस फिगर में दिख रहा है कि ए सब यूनिट अलग कर दिया है, देन बी सब यूनिट अलग कर दिया है, देन दे क्रिएट अ कॉम्बिनेशन विद द हेल्प ऑफ डाइसल्फाइड ब्रिज एंड क्रिएट एन एक्टिव इंसुलिन एज एन human insulin. Okay, the chain A and chain B were produced separately over here, extracted and combined by creating a disulfide bond to form a human insulin. इससे क्या होगा कि इंसुलिन ये एज इट इज जैसा हमारा ह्यूमन इंसुलिन है उसके जैसा ही रहेगा. नो एनी एलर्जिक इज देयर, no any other reaction is there, no any other side effect is there, and we produce them in a bulk. Okay, and for that we do not require any killing of any slaughter animals like a cattle and pigs. हमें उसको मारने की भी ज़रूरत नहीं रहेगी and we create a same insulin as found in a human beings. So this one a major application of a biotechnology, major role of biotechnology in the field of medicine. This one is the great example that show how biotechnology that is helpful for our medicinal point of view. I hope students आज को आपके इस वीडियो में समझ आया होगा. इन अवर नेक्स्ट वीडियो हम बात करेंगे हमारे नेक्स्ट बायोलॉजिकल एप्लीकेशंस की, दैट इज द जीन थेरापी एंड मोलिकुलर डायग्नोस्टिक. तब तक आप इसको रिवाइज करते रहिए. थैंक यू एंड टेक केयर.